Welcome to Lecture 10 on the topic of carbohydrate partitioning. This lecture is delivered across 3 parts, and this is the second part where we will concentrate on the physiological and ecological considerations of carbohydrate partitioning. This lecture is part of the subject Plant Physiology, which is a component of the Bachelor of Agriculture and Technology, a degree jointly offered by Melbourne Polytechnic and La Trobe University. For further information on this course and other courses that we offer, please visit our website at www.melbournepolytechnic.edu.au. My name is Dr. Nikki Cooley. In this subject, you are learning about plant physiology by taking a virtual journey through the plant. We are up to Lecture 10. Please ensure that you have watched all the lectures and completed the Moodle quizzes before continuing with this lecture. It is particularly important that you have listened and understood Part 1 of the Lecture 10, as this lays down many of the concepts that you need to familiarise yourself in order to understand Part 2. In Lecture 10, Part 2, we'll be concentrating on both the photosynthetic light reactions, photosynthetic carbon reactions, and the consequence of the products of these reactions, carbohydrate partitioning, and how abiotic impacts can influence the physiology of these 3 important processes. I will be introducing to you the concept of limiting factors, and we will look at some of these limiting factors. The first one we will look at in detail will be covered in this lecture, light. The others, temperature and carbon dioxide, will be covered in Part 3. Physiology is the scientific study of the function of living systems. This is characteristic of the normal functioning of a living organism, while ecology is the science of the relationship between these organisms and their environments. The impact of the environment on photosynthesis is of interest to plant physiologists, ecologists, and agronomists. In order to understand this, you need to understand the physiological responses of the photosynthesis to environmental factors such as light, carbon dioxide concentrations, temperature, and humidity. Important to you as either an agronomist, a viticulturist, or a plant physiologist, plant productivity is based on crop yields, which in turn is very dependent on photosynthetic rates. A limiting factor is a factor or condition that either temporarily or permanently impedes photosynthesis. In 1905, it was Beckmann who hypothesized that the rate of photosynthesis is limited by the slowest step in the process, and he termed this the limiting factor. So how many environmental factors can limit photosynthesis at any one time? It appears that there are 3 major metabolic processes which are important for optimal photosynthetic rates. These are the rubisco activity, the regeneration of RUBP, and the metabolism of triophosphates. These activities describe the demand for carbon dioxide as a substrate. However, the actual rate of carbon dioxide supply to the cell is controlled by the stomatal guard cells. Thus, the supply and demand functions take place in different cells. The coordinated actions of demand by photosynthetic cells and supply by stomata determine the leaf's photosynthetic rates as measured by the net carbon dioxide intake. The figure below shows the structural dimensions of a leaf. The figure below, if magnified, demonstrates 2 different structures of a leaf. One leaf was grown in the shade and one leaf was grown in the sun. You can see some of the components which are different, such as the diameter and the complexity of the spongy mesophyll. So let's look at some of the primary functions of leaves. How does the leaf anatomy and structure control the absorption of light? The rate of photosynthesis is limited by different factors. For example, inadequate light or carbon dioxide, or excessive light. There are limits on the levels of control a plant has over photosynthesis, thus limits to acclimation to sun, shade, high and low temperatures, and degrees of water stress. Differences in the amount and the type of light means we need a uniform way of measuring and expressing the light that impacts on photosynthesis. So let's look at measuring light in a little bit more detail. Light reaching the plant is a flux, which can be measured in either energy or photon units. Irradiance is the amount of energy that falls on a flat sensor of known area per unit time. Watts per square meter. Remember that time is in the term watt, therefore 1 watt equals 1 joule per second. Photoirradiance is the number of incident quanta striking the leaf, in moles per meter square per second, where moles refers to the number of photons of light. This means that 1 mole of light equals 6.02 times 1023 photons. Quanta and energy units can be connected provided the wavelength of light, known as lambda, is known. The photon energy is related to its wavelength. Where c is the speed of light, h is the Planck's constant, and lambda is the wavelength of light. Once you have all of this information, you are able to calculate the photon energy of light. So what does all this mean? Well, it means that a photon of light at 400 nanometers has twice the energy of a photon of light at 800 nanometers. And this relationship is very important to plants. So why are we learning about the characteristics of light? Well, plants perceive and require certain energies at certain wavelengths. We have developed what is known as the photosynthetic active radiation, or PAR. This is a measurement of light between 400 and 700 nanometers, and it is normally expressed as a quanta. That is a measure of moles per meter square per second, and then a measure of the concentration of energy over the distance that energy applied over time. Incoming sunlight will strike a leaf surface at a variety of angles, as you can see from the figure below. When sunlight deviates from directly over the leaf, or what we call perpendicular, irradiance is proportional to the cosine of the angle of the light rays. Now that we understand the relationship of the angle of light and a leaf surface, we can begin to ascertain how much light is on a sunny day. So the photosynthetic active radiation, or PAR irradiance, is approximately 2000 micromoles per meter squared per second at the top of a dense canopy. This is approximately 10 micromoles per meter squared per second at the bottom of a dense canopy. This is quite a difference, and the shading of the leaves reduces this incident radiation. About 1.3 kilowatts per meter squared of radiant energy reaches the Earth. This is less than 5% ultimately converted into carbohydrates by leaves. So 50% of the incident light is of a wavelength either too short or too long to be absorbed by the photosynthetic pigments. Of the photosynthetically active radiation that is absorbed, about 15% is reflected or transmitted through a green leaf. That means it either bounces off the leaf, is absorbed in the middle of the leaf, or travels all the way through the leaf without being used. You will remember from Lecture 6 that light is absorbed in the chlorophyll structure and that this is wavelength specific. 680 and 700 nanometers were the reaction centers for photosystem 2 and photosystem 1. Blue light is also absorbed in the chlorophyll pigments. Therefore, chlorophyll absorbs very strongly in the blue and the red regions of the spectrum, and thus transmitted and reflected light are vastly enriched in green. You can see a visual representation of this in figure 9.3 on the slide. Of the 85% photosynthetic active radiation absorbed, significant amount lost as heat, so less than 5% of the incident light is converted to carbohydrates. So leaf anatomy plays an important part in maximizing the absorption of light. Leaves are highly specialized for light absorption. The epidermis is transparent to visible light and cells are convex. Convex cells act like lenses, thus focusing light so the intensity reaching the chloroplast is many times greater. The palisade cells are shaped like pillars that stand in the parallel columns, 1, 2, 3 layers deep. More light than expected penetrates the first layer due to the sieve effect and the light challenging effect. The sieve effect comes about as chlorophyll is not evenly distributed throughout the cell. The packaging of chlorophyll results in shading and creates gaps between chlorophylls where light is not absorbed. Light channeling occurs when some of the incident light is propagated through the vacuoles of palisade cells and through the air between the cells. The spongy mesophyll cells between the palisades are very irregular in shape and are surrounded by large air spaces. This generates many interfaces between air and water which will reflect and refract the light. This has the consequence of randomizing the direction of light travel, interface light scattering. The strategies that we have just talked about are strategies that enable plants to optimize light and the use of light. However, there are some environments, for example deserts, where there is much light around, and this can be potentially harmful to the leaves. These plants have leaves where special anatomical features occur. They have hairs, they can have salt glands, and they can have an epicutical wax layer. All of these characteristics increase reflection of light, thus reducing light absorption. Light absorption can be decreased up to 40%, minimizing heating and other problems. Some of these environments are found often here in Australia, and an adaptation to the native Australian plants to the high light environment is very common. In many habitats, a lack of light is a problem. Leaves can shade each other, and the leaves at the lower levels experience different light quality, and thus they have lower photosynthetic rate. Trees represent an outstanding adaptation for light interception. Elaborate branching vastly increases this. Very little photosynthetic active radiation penetrates the bottom of forest canopies, and this shows some of the efficiencies that these canopies have obtained. In shady habitats, sunflecks are a common environmental feature. These are patches of sunlight that pass through small gaps in the leaf canopy. Sunflecks can arrange the photon flux incident on a leaf more than 10-fold within seconds. Critical energy available for only a few minutes. For some leaves, these photons contain nearly 50% of the total light energy available. Also, they play a role in carbon metabolism of lower leaves in dense canopies. Rapid response by photosynthetic apparatus and stomata are of substantial interest due to the mechanisms for capturing short bursts of energy. So how do leaves influence the light levels within a canopy? Well, the angle of the leaf determines the amount of light that leaf receives. Leaves that are horizontal to the sun can receive more light. However, leaves exposed to full sun tend to have steeper leaf angles, so less than the maximum light is incident light. This allows more light to penetrate into the canopy. Leaf angles tend to decrease with increasing depth. Some plants control light absorption by solar tracking, and this was first observed in sunflowers. Leaves continuously adjust orientation, remaining perpendicular to the sun's rays. This includes many species such as alfalfa, cotton, soybean, and lupin. Some plants can develop in response to a range of light regimes, and this process is called acclimation. A growth process in which each new leaf has a set of features suited to its environment. Importantly, the ability given that shady habitats receive less than 20% of PAR and deep shade less than 1% of PAR. Leaves that develop under very sunny or shady habitats often are unable to persist when transferred. Sun and shade leaves have contrasting biological features. Shade leaves have more total chlorophyll per reaction center. They have reaction ratio of chlorophyll B to A. This produces a ratio of 3:1 of photosystem 2 reaction centers. They can add more antenna chlorophyll to photosystem 2 to increase absorption and tend to be thinner. While sun leaves have more rubisco and a larger pool of xylophil cycle compounds. They are thicker and they have longer palisade cells. This is too light. You can measure net carbon dioxide fixation in leaves across varying levels of light, and this allows us to construct light response curves, i.e. the impact of the photosynthetic rate or the ability for photosynthesis to be efficient against the amount of light absorbed. On the figure on the slide, you can see that the photosynthetic rate is plotted against the absorbed light. You will notice in the first stage of the reaction on the left-hand side that the response is linear. That is, as the light increases, so does the photosynthetic rate. This component, or the light yellow component of the graph, is light limited. Then you reach a point where there is enough light for you to optimize the photosynthetic capacity. The reaction then is known as carbon dioxide limited. In this species, the light limiting is about 300 micromoles per meter squared per second. So let us look into a little bit more detail about the photosynthetic responses to light. In the dark, as we have learnt in lectures 6 and 7, in C3 plants there is no photosynthetic carbon assimilation. But mitochondrial respiration continues, and so you get carbon dioxide released from the leaves. At greater photon flux levels, or higher light levels, CO2 assimilation reaches a point at which carbon dioxide uptake exactly balances carbon dioxide release. This is known as the light compensation point. This point varies between plants grown in the shade and plants grown in the sun. For example, the light compensation point for shade plants is 1 to 5 millimolars per meter squared per second, whereas the light compensation point for sun plants is somewhere between 10 to 20 millimoles per meter squared per second. Lower for the shade plants as respiration rates are lower. Hence, shade plants survive in light-limited environments as they achieve positive carbon dioxide uptake rates at lower values of photosynthetic active radiation than sun plants. Another measurement of photosynthetic efficiency is called the maximum quantum yield. It describes the linear relationship which persists at light levels above the light compensation point. Photosynthesis here is light limited. More light stimulates more photosynthetic systems. This slope reveals the maximum quantum yield of photosystem. In C3 plants, the reduction from the maximum quantum yield is caused by energy loss through photorespiration. In C4 plants, and you'll remember the mechanism is different to C3 plants, reduction of quantum yield is caused by the additional energy requirements of carbon dioxide concentrating mechanism. The quantum yield also varies with the temperature and the carbon dioxide concentration because of their effect on the ratio of carboxylase to oxygenase reactions of Rubisco. How C3 and C4 plants respond to the quantum yield and leaf temperature are illustrated in this figure. In situations where higher photofluxes occur, the photosynthetic response to light starts to level off. At the point where it starts to level off, we call this the saturation point. This actually indicates that light is no longer limiting and that something else has become limiting in the photosynthetic process. Examples of other components that can become limiting are the electron transport rate, the Rubisco activity, or the metabolism of triose phosphates. Above saturation point, photosynthesis is referred to as carbon dioxide limited. In situations where plants are exposed to excess light energy, there are strategies employed by the leaves which enable them to get rid of or dissipate the excess energy, and this stops the plant being exposed to damage to the photosynthetic apparatus. Most important mechanisms involve the transfer of absorbed light energy away from the electron transport, so the energy then is converted into heat. The xylophil cycle is important for heat dissipation. An alternative method for reducing excess light energy is to move the chloroplasts so that this excess energy is no longer in contact with the chloroplasts, and therefore they are not exposed to the damage. In the experiment you can see images of chloroplasts that have evolved under the darkness, weak blue light and strong blue light, and you can actually see these moving here. With excess light energy, the reaction centre of photosystem 2 is inactivated and often damaged, and this is called photoinhibition. There are two types of photoinhibition, dynamic and chronic photoinhibition. Dynamic photoinhibition operates under moderate excess light, decreases in quantum efficiency, but maximum photosynthetic rate remains unchanged. It's caused by the diversion of energy to heat. Chronic photoinhibition is where excess light occurs. This damages the photosystem and decreases both maximum photosynthetic rate and quantum efficiency. This is long-lasting and affects over weeks or months. The associated reading with this chapter will be given in part 3. So let's summarise what we have learned. Well, we have introduced the physiological components of photosynthesis and the concept of limiting factors. We have looked at the abiotic input light in some detail. You will have learnt by now the relationship of light and photosynthesis and just how important it is and what that role is. You are aware of the measurement of light and how this can be interpreted. The leaf anatomical characteristics and how they play and use light have also been examined, both here and in the DIY practicals. We have learnt that there are different types of leaves with respect to how they handle light, that is sun and shade. That there is a process called acclimation, which enables plants to adjust to light. There is a concept called saturation point, which we have also covered in this topic, as well as the important component of photoinhibition. This can reduce productivity for plants if too much photoinhibition occurs. This is the end of part 2 of this lecture on carbohydrate partitioning, physiology and ecological considerations.